So I'd now like to call up to the podium Susheel, Bhanu, Busi, who's going to talk about adenoma susceptibility in colon cancer.
Good afternoon. First, I'd like to thank the organizers, GSA, and IMGS for the travel award that afforded me this immense learning experience. Also, at the outset, my apologies because I speak Her Majesty's English in my own Indian way. Colorectal cancer initiation, progression, and development can be influenced by multiple factors. Just a few: genetic predisposition, age, sex, lifestyle, underlying conditions, epigenetics, and immune responses. Lately, there's been accumulating evidence that the microbiome has a significant effect on these factors and the disease itself. However, several of the human studies that have been published in light of this have mostly been retrospective, always showing associations between the presence or absence of tumors with certain bacterial species. In fact, quite a few of these studies showing these associations have never looked at the precancerous microbiota, the progression of cancer itself, and the dynamic microbiota changes. In order to understand the effect of the microbiome on colorectal cancer, we use a genetic rat model of human familial adenomatous polyposis. The rat itself, the polyposis in rat colon, or the Pirc rat, was developed by Drs. Amos Landgraf and Kwong in the lab of Dr. William Dove at University of Wisconsin-Madison by E&U mutagenesis. The strain carries a single nucleotide variant in the APC gene, which was induced by E&U, and this affects the Wnt signaling pathway. The Pirc rat recapitulates the human phenotype, wherein you see colonic adenomas, as seen in the colonoscopy images on the right, and it also is orthologous to the human FAP condition. We also see a significant number of tumors in males compared to the females, recapitulating again the human phenotype that is observed. Using this Pirc rat model to determine how variable complex gut microbiota affect disease susceptibility, we used a strategy called complex microbiota targeted rederivation, or CMTR. In this strategy, we crossed a male heterozygous Pirc rat with a wild-type inbred female rat, and the embryos thus generated were then collected and rederived using 3 surrogate dams, a SD, a Fisher, and a Lewis from 2 different vendors. The pups were allowed to be naturally delivered by the dams, thereby obtaining genetically identical Pirc rats, however, carrying different and distinct microbiomes. We then used longitudinal colonoscopies, starting at 1 month of age, and also collected fecal samples from these pups over time. Going forward, I'll refer to these 3 groups as the SD, the Fisher, or the Lewis group. The fecal samples that we collected from the pups and the surrogate dams at different time points, we extracted bacterial DNA from these, and then using the Illumina MySeq platform, we identified the operational taxonomic units, or OTUs, or the bacterial taxa that are found in these particular groups. We annotated the sequences based on the Green Genes database to known OTUs, and on the right here you see a representation of these sequences down to the species level. If you were like me, you were probably looking at Family S247 because I like the color blue. When we then looked at the surrogate dams to assess how their microbiomes differed, we found that all the 3 surrogate dams carried a distinct microbiome from each other. Interestingly, though the Fisher and the Lewis rats came from the same vendor, they carried a different microbiome themselves. What was really curious to us was the fact that the Lewis microbiome was significantly different to the other 2 groups, where it had this really high prevalence of Prevotella copri. After this, we looked at the pups themselves, the CMTR pups, to see how well they resembled the dams. And here is just a representation of a Fisher and a Lewis dam with one of its pups at different time points. The y-axis shows you the relative abundance of the OTUs, each color here representing a single OTU again. And we noticed that by 1.5 months, all the pups resembled their respective dams. And this was prior to disease onset, which was judged by colonoscopy videos. To determine what these differences in the microbiome had on the disease phenotype itself, we analyzed tumor counts in these animals after they were sacrificed at 6 months of age. We noticed that there was a significant decrease of tumors in the Lewis groups, both in the males and the females. What was really interesting, was these 2 right here, a male and a female in the Lewis microbiome profile that did not develop any colonic tumors. This was the first time in over 500 Pirc rats to date that we saw an incomplete penetrance of the phenotype. Now, to understand what microbiome differences may have caused this difference in phenotype, we did a principal component analysis where each triangle here represents a single male rat at 6 months of age, basically showing its entire GM profile. The closer the triangles are to each other, the more similar they are. The distant, the more dissimilar. We noticed that there was a significant difference between the Lewis GM rats compared to the SD and the Fishers. On the right, you are seeing a loading plot, which basically shows you a few OTU or taxa that we think possibly contribute to this variation that is observed within the GMs. In order next to understand how the GMs were possibly creating this phenotype, we looked at individual bacteria and their correlation with the tumor abundances. When we looked at the OTU relative abundance at the weaning time point and compared it with the tumor counts from 6 months, here you're seeing the relative abundance on the y-axis and the tumor counts on the x. You will notice that certain bacteria, especially the ones listed here among others, showed a positive correlation between the relative abundance of the taxa and tumor counts themselves, basically indicating that these OTUs possibly have an effect as far as tumor incidence is concerned. We also found other bacteria like Prevotella copri and Disulfo vibrio species as being significantly different between the groups. And owing to the fact that Disulfo vibrio species and P. copri have different metabolic functions, we used what is called a phylogenetic investigation of communities by reconstruction of unobserved states, or PICRUSt to be short. This was developed by the Huttenhauer lab, and what it does is it takes a look at the GM and predicts the metabolic function of the microbiome profiles itself. And when we did this, we noticed that there were 2 pathways that came up that were significantly different between the groups. And these pathways were related to bacterial hydrogen sulfide genes. Specifically, they were the sulfate transport system and the sulfur reduction to hydrogen sulfide. Owing to the genotoxic nature of hydrogen sulfide, we then used a pyrosequencing assay specific to the Pirc allele, and we noticed that the SD rats and the Lewis rats showed a significant loss of heterozygosity, specifically 100% and a 96%. Again, indicating here that maybe the pathway in which tumors are initiated in the Fisher rats could be different compared to the other groups. In conclusion, we saw that the complex gut microbiome modulates adenoma susceptibility, where the Fisher Pirc rats, which carried the Lewis GM, was distinct from the other complex microbiota and showed, for the first time, an incomplete penetrance of the disease. There were certain microbial compositions and taxa that correlated positively with tumor multiplicity. And using the PICRUSt analysis, we noticed elevated H2S-related bacterial genes, especially in the SD and the Lewis GMs, which possibly correlates with the loss of heterozygosity that was observed in the same GMs as well. Last but not the least, there may be a potential association between the variable complex gut microbiota and the loss of the wild-type APC allele. With that, I would like to thank my mentor, Dr. James Amos Landgraf, my lab members, both former and current, Doctors Breider, Erickson, and Franklin, and everybody on the right for their contribution, the GSA, the IMGS, and everybody here for your presence. Last but not the least, if you'd like to learn about CMTR, please talk to one of our lab members who are all present here at the meeting. Thank you.
Thanks for a great talk. Do we have any questions for...
So you look for a positive correlation between tumor number and a particular species. Did you also look for a inverse correlation or negative correlation?
Yes, we did, and I forgot to mention that we did find a few OTUs that were inversely correlated as well, which I did not mention in this, and they were spread across the 3 GMs.
So in theory, the microbiota could affect tumor development in 2 broad ways. One, directly through breaching epithelial barriers and causing tissue damage, and indirectly by educating the immune system.
Yes.
So do you have a point of view about that based on your data to date?
Just based on the data at this point, we could be the genotoxic nature that probably is bringing in an immune response, actually, responding to this nature of the genotoxicity. But it could, like you mentioned in your talk, it could be this combination of multiple systems responding to the tumors themselves.
So did you do any blood immunology?
With these rats, we did not. We just did a full CBC count. We didn't really look at them at this point.
And you talked about tumour burden, but what about the histology? Did that change at all?
No. To be honest, we did a cursory look at the histology across all the GMs, but we did not really see upfront any difference in grade of the tumors, and most of these only develop adenomas anyways, the model itself.
And did you leave the mice that didn't develop any tumours longer to see whether it was just a latency or...?
No, we sacrificed all of them at 6 months, so we didn't have that opportunity to take them out.
So if there's no more questions, please thank Susheel for a great talk.
Thank you.